Salvete Discipuli. This is Mix Martin with a recap of lessons 7 through 9. Cell biology lesson 7 is about animal and plant cells, and the key question is, what are the differences between plant and animal cells? The objective is Discipuli will be able to draw diagrams of plant and animal cells. The first section is about comparing plant and animal cells. This is a drawing for your reference. Right now, please go to your notes. Check what organelles are found in both plant and animal cells. And then also answer what organelles are found only in plant cells. So pause the video and do that now. Hopefully you identified that plant cells have a large central vacuole. Those descriptors are important. Plants have a large central vacuole. That matters because there are vacuoles sometimes in animal cells, but only plants have this large central vacuole. Plant cells also have a cell wall, which is this green structure surrounding the cell. It keeps plants rigid, holds them up. Obviously, plants don't have a skeleton like you do, so the rigidity of the cell wall and the water pressure of the large central vacuole keeps them stiff and rigid. These are chloroplasts. Plant cells also have chloroplasts. Structures that we can see in both plant and animal cells include the nucleus, the endoplasmic reticulum, which is smooth endoplasmic reticulum, as well as rough endoplasmic reticulum that's studded with ribosomes, the Golgi apparatus, mitochondria, and then we can call this the cytoplasm. It's the region outside of the nucleus. Or if we want to be specific about the fluid, it's the cytosol. Going on to eukaryotic cell drawings. So you should be able to produce a drawing of a plant cell and an animal cell. And there are full videos of me drawing both of these with a document camera on this YouTube channel. So this image shows a plant cell. Excuse me, this is an animal cell. It's an animal cell. I can tell it's an animal cell because it doesn't have a cell wall, it doesn't have a large central vacuole, and it doesn't have any chloroplasts. Look at the structures that it does have. I'm going to highlight a few things. The nucleus has a double membrane with these nuclear pores. So the nuclear pores are essential because they allow mRNA molecules to leave the nucleus. If you draw this without the double membrane, you definitely won't get the point. So here's the inner membrane, here's the outer membrane, and then the space between them, that's the nuclear pores. This region is called the nucleolus. It's where the RNA that's part of ribosomes is synthesized. We have the rough ER with the ribosomes. We have smooth ER, no ribosomes. The Golgi apparatus is like a stack of flattened pancakes, and we can see some vesicles branching off of it. So the Golgi apparatus, you should remember, modifies proteins coming from the rough ER, and oftentimes those proteins are to be exported, so that's what's going on here. This is a vesicle that's moving towards the plasma membrane, fusing with it and expelling its contents. This is a lysosome, contains digestive enzymes to break down whatever it fuses with. And I think I got everything. For the mitochondria, please make sure that you're drawing two membranes and that the inner membrane has these infoldings called cristae. So without those infoldings, you haven't clearly represented a mitochondrion. Please remember that there are ribosomes as well. And these ribosomes are ADS because this is a eukaryotic cell. Here's our animal cell. And again, we can recognize the large central vacuole. Please include those adjectives. We can recognize chloroplasts with those stacks of thylakoids. And our cell wall, we have here the thicker layer that's outside of the plasma membrane. Don't forget that plant cells have both a cell wall and a cell membrane. Plasma membrane and cell membrane are the same thing. And then the other structures we also saw in the animal cell, so we don't need to go over those. Going on to lesson 8, which is about microscopy. And the question is, what do cells look like under a microscope? Discipuli will be able to distinguish between electron microscopes and light microscopes and identify organelles in electron micrographs. So the first section is about microscopy. Light microscopes are what you may have used in a science classroom before. We have a lab coming up where we'll get to know them and use them. So they use a series of lenses to magnify a specimen. They're basically like a double magnifying glass. So there's a lens here, and then this is a rotating piece that has 3 different lenses. You only look through one at a time, but it's the magnification of this lens times the magnification of whatever lens you have here. These can magnify a specimen up to 400 times, so they can make it look 400 times bigger than it is in real life. These cost a few $100s usually. They don't take any training to use besides a few minutes of learning how to focus. And a cool advantage is that you can look at living specimens. So when we saw the video of the paramecia earlier, that was taken through a light microscope. And those living paramecia were just swimming in the water on the microscope slide, and we were able to see them do some of their normal behaviors. These are some leaf cells. So this is one cell. Within it are the visible chloroplasts. So the small green things, that's not a cell. This that I'm circling is a cell. And the small green things are the chloroplasts. That's another advantage of light microscopes. You can see images in color, so you can see the actual color of these specimens. This is a picture of human cheek cells under the microscope. They've been stained, so they're not naturally that bright pink, but it makes it easier to see. If you take a toothpick and you just gently scrape the inside of your cheek, you can make a microscope slide and actually look at those cells under the microscope. So we talked about light microscopes. There are also electron microscopes. They can magnify images up to 10 million times. So these are much more powerful. They take up much more space. They cost tens of thousands to even millions of dollars. It takes special training to know how to use them. Most universities will have these, but it's not something you would find in a high school. These are some micrographs taken with an electron microscope. So this is a tardigrade or a water bear. This is just an ant. And then this is a mitochondrion. So we can see that the inner membrane has these infoldings, and that's how we recognize it as a mitochondrion. There are 2 types of electron microscopes. There's scanning electron microscopes and transmission electron microscopes. So scanning electron microscopes can give us this sort of 3D picture. So that's the water bear and the ant. For transmission electron microscopes, we have to cut a very, very thin slice of our specimen, and the electrons actually pass through the specimen, and that's why they don't make a 3D image. It's just kind of a flat image. In both cases, in both kinds of microscopes, I should say, the specimen can't be alive. It has to be dead because it's prepared in a special way so that the electrons interact with it in a way that the microscope can detect and understand. So for scanning, you basically fire a beam of electrons at the specimen and measure how they bounce back. That's what the electron microscope detects. And for transmission, you're actually sending electrons through the specimen and measuring on the other side how they get deflected by the specimen. So that's why I said for transmission, you have to have a very thin slice of your specimen. You need to know this language for IB. It says electron microscopes have a greater resolution than light microscopes, and that means you can see much smaller structures. Resolution is the smallest distance between 2 points on a specimen that can still be distinguished as 2 separate entities. So if you took an image from a light microscope, if I go back, we could blow this up, we could make it bigger and bigger and bigger, but it's just going to get blurry. So we can't increase the resolution of a photo after it's been taken. And that's why it's important to know that electron microscopes have this greater resolution.